Hi there, I'm Gabby.
Hello, I'm Monica, and in this video we'll be discussing our article which is titled hnRNPD, an understudied subfamily affected in sporadic TDP-43 proteinopathies.
Amyotrophic lateral sclerosis and frontotemporal lobar degeneration are two devastating neurodegenerative diseases which sit on two ends of the same disease spectrum. TDP-43 is a member of the heterogeneous nuclear ribonucleoprotein, or hnRNP family, which plays a critical role in regulating RNA processing. The nuclear depletion and abnormal aggregation of phosphorylated TDP-43 in neurons is a hallmark of ALS and FTLD. Evidence suggests that the nuclear depletion and cytoplasmic aggregation of pTDP-43 could drive neurodegeneration in ALS and FTLD by disrupting RNA processing, including by the loss of TDP repression of cryptic splicing. Other members of the hnRNP family, such as hnRNPA1, C, and H, have been shown to modulate TDP-43 function and abnormal splicing events, although few studies have investigated whether hnRNPs are affected in sporadic TDP-43 proteinopathies, especially using patient tissues. Therefore, using immunohistochemistry and immunofluorescence, our study assessed 7 hnRNPs, all which have various important roles in RNA processing. The knockout of a single one of these hnRNPs can result in the differential splicing of thousands of RNA transcripts.
The hnRNPs we specifically assessed were hnRNPA1, A0, and A2B1 from the AB family, hnRNPC, and then DL and AB from the hnRNPD family, and hnRNPH. We assessed them within the frontal and motor cortex, which are two regions involved in the early and late stages of TDP-43 pathogenesis. Cases included sporadic ALS, ALS-FTLD, and FTLDN, which is based on the TDP-43 classification of long dystrophic neurites, and we also looked at FTLD-NCI for cases characterized by neuronal cytoplasmic inclusions. Using immunohistochemistry on cortical regions from ALS and FTLD, our results showed distinct patterns of nuclear localization and depletion across ALS and FTLD cases. Firstly, in ALS, we observed normal nuclear localization of the hnRNPA-B, hnRNPC, and hnRNPD, and H2 families in the motor cortex.
However, in FTLD, we found nuclear depletion of hnRNPA1, hnRNPC, hnRNPA-B, and hnRNPDL in the frontal cortex, with some cytoplasmic accumulation of hnRNPA1 and C. Notably, there was a significantly greater loss of nuclear hnRNPD in all FTLD groups compared to ALS, and a significant loss of nuclear hnRNPA1 and C in FTLD ALS and FTLD NCI groups compared to ALS.
Next, we performed correlation analysis to understand any relationships between the different hnRNPs and pTDP-43. Firstly, we found a strong relationship between nuclear hnRNPC and hnRNPA1, and a significant nuclear loss in the hnRNPD family was associated with an increased cytoplasmic pTDP-43, which highlights an understudied subfamily in sporadic TDP-43 proteinopathies.
We then used immunofluorescence to determine whether these hnRNPs with nuclear depletion in FTLD cases are co-localizing with cytoplasmic pTDP-43. We did not find co-occurrence between cytoplasmic hnRNPA1 and pTDP-43 inclusions. Interestingly, cytoplasmic hnRNPC was observed in neurons without cytoplasmic pTDP-43, and only rare neurons showed co-localization of cytoplasmic hnRNPC and pTDP-43.
In conclusion, we suggest that the nuclear loss of hnRNPD in FTLD cases and its significant relationship with cytoplasmic pTDP-43 may suggest a regulatory interaction between proteins. So both hnRNPDL and hnRNPA-B have important roles in neuronal function, with hnRNPDL modulating the splicing of cytoskeleton and synapse genes, and then hnRNPA-B playing a role in neuronal survival and glutamate signaling, which are both impaired in FTLD. Meanwhile, the significant nuclear loss of hnRNPC in FTLD cases with cytoplasmic hnRNPC inclusions, but not with TDP-43 aggregates, rather points to independent pathways in FTLD. hnRNPD and hnRNPC can be detected in plasma samples, and they're reduced in patients with bulbar compared to limb onset ALS, which suggests a need for further investigation in their potential as proximal biomarkers of TDP-43 dysfunction or disease subtypes, particularly as hnRNPC is already recognized as a promising biomarker in cancers. hnRNPA1, which was reduced in the nucleus of neurons in FTLD patients, has been shown to be a druggable target in various cancers where it is also aberrantly localized and expressed. So future research may determine whether hnRNPA1 targeting compounds would also be appropriate for TDP-43 proteinopathies.
Overall, we have shown that these hnRNPs, all with important roles in RNA processing, are aberrantly expressed in neurons of patients with FTLD. This suggests the involvement of widespread missplicing events in the pathogenesis of sporadic FTLD and underscores the need for further investigation into the role of hnRNPs in disease pathogenesis across the ALS-FTLD continuum, using experimental models to explore mechanistic involvement, and to investigate their potential as modulatory targets or markers of TDP-43 dysfunction. We would like to thank the brain donors and their families for their generous gift to research, the Sydney Brain Bank and our supervisor Rachel Tan and all the researchers involved in helping with this research. Thank you for listening.
Thank you.